and also carcinogenesis, right? I remember last few lectures ago, some students asked about the difference between all these terms, right? So today we want to really explore and understand why we actually classify a certain things as genotoxic, and then sometimes we talk about teratogenesis, and then sometimes we say something is mutagen and something is carcinogen. So what are the differences? So today we want to learn a bit, okay, on why we need to do this kind of classification. Right, so yeah, so far we have come to week 7. Yeah, we have covered all those basic things about dose response relationship, and then toxicokinetics, toxicodynamics on how pollutants or toxicant can enter into our body, and then what kind of effect it can exert. And then after that, we also look into something that is more chemistry, which is the how this toxicant can move around in the environment. So today we look into something that is much more interesting, right? We look into how those toxicants can actually affect our genes, right? So we are going to start off with something that is very basic, okay, in terms of how DNA is in our cell, and then after that we look into how this DNA can divide either through meiosis or mitosis, right? And then after that we are looking into how this toxicant can actually affect the process, and then leads to the carcinogenic effect, right? So next week we want to go back to the chemistry part a bit. We look into something that is more practical, right? We want to estimate the toxicity of a certain toxicant or pollutant using this method, test method, right? So for week 8 we will be doing something like let's say for my case, right, in my study I look into a pollutant, and then this pollutant can be degraded into many different kind of intermediates. So each of these intermediates, they are basically toxic, right? So how we can estimate the toxicity of these intermediates, right? We are going to explore that, right? So at the end of the lecture, I will give you a link, and this link leads to the software whereby you can download, and next week we are going to practice a bit on how we can actually do this toxicity estimation, right? So today's lecture will be on understanding the differences, right, between genotoxicity, teratogenesis, mutagenesis, carcinogenesis, and then we also want to be able to describe all these terms. And finally, we want to talk about the basic tests, right? What are the basic tests involved when you want to investigate all those different toxicity, like genotoxicity, teratogenesis, and so on, right? Yeah, before I forget, next week, as I mentioned previously, we will have an assignment, right? So I will push the assignment date to early next week, maybe on Monday. Okay, so Monday I will upload the questions. I will let Dr. Fitri know that once I have uploaded, and then after that you guys can download it, okay, and then after that try to solve the problem, and the due will be by 1 November, right? Okay, so let's look through these molecules of life, right? When we talk about how all those carcinogens, mutagens can really do some difference, can make some difference in our body. We're actually talking about how they can induce changes to our DNA. Okay, most of the time they are going to affect our DNA, and when this DNA is affected, right, sometimes it can change how the DNA is translated into proteins, right? In this case, it will cause many effects. For instance, you can have excessive growth of a cell, okay, which means your cell will grow uncontrollably, right? And then you will see that it forms tumors, and then tumors will spread all over the place. Okay? And sometimes you can have the toxicant causing change to your reproductive system. Okay? Sometimes the sperms and the ovum, it can be affected, and then after that the offspring will be affected, right? So it depends on what are the toxicants and how they can affect. Okay? And in other words, we are looking into the mechanism of how the toxicant can affect the DNA, right? So in this DNA and RNA, right, they actually replicate, right? So there's something called replication that is happening in our body every day. right? Which means that you can see the cell is dividing, okay, either for growing, okay, or for sustaining our body, right? So during this growing, we actually see the DNA being duplicated, right? So in other words, all those cells that we have in our body, they have the same DNA information, right? So this is the wonder of our body, right? Because we can actually have a mechanism, right, which involve a lot of enzyme, right? So all those enzyme comes together, and then they start to carry out certain reactions that enables the DNA to be transformed, okay, from this DNA, okay, into another one DNA, right? So there's a duplication of DNA due to this complex reaction, right? So because the reaction is very complex, right? So many different kind of biochemical reaction can happen. We see that a lot of things can go wrong. Okay, as long as one toxicant can affect one process, then you see the replication process, the transcription process, the translation process will be affected, right? So I'm sure most of you have heard of this term, transcription and translation, in your early years, okay, of your study. These two, they are concerning the expression of the information of the DNA, right? So generally, this is how it starts. You have a DNA, right, which can replicate, right? So you will continuously have your DNA being produced in your body for different cells, right? And then after that, you have transcription, okay, which means that your DNA is being copied by an RNA so that this RNA can move out of the nucleus. So this DNA is always in the nucleus, right? And then to make this DNA go out from the nucleus, it has to be in the form of RNA, right? So that's why we can see DNA will be transcribed to RNA, right? So this RNA will then be going out from the nucleus. Okay? It will go into the cytoplasm, and then after that, you will have certain process happening. Okay? The ribosome will be attached to the RNA, and then after that, you have the protein being synthesized in the cell itself. Right. So there are many different kind of protein functions in our body, which these are the proteins that are very important in our body, which will be produced by DNA. Okay? And sometimes they help in terms of the transport of chemicals. For instance, if we talk about antibodies, these are the things that can be produced by the so-called DNA translation, okay, antibody. Okay, like for example, if you are sick, right? So your bodies will produce more antibodies. So how this cell produce antibody depends on the signal that they receive. Okay. So when they have the signal from the DNA or from the RNA or specific things, right, then you will start to produce all those antibodies. For instance, if we look at the vaccine, Pfizer, right? Pfizer vaccine, it is based on the RNA technology, which means that you inject something and then it will have RNA which goes into your cell and then starts to produce the antibodies. Right, then we also have receptor. Okay, and then receptor, they are the one that is going to recognize what kind of chemicals, proteins going into the body. Then sometimes your RNA can be translated to enzymes. Okay, this one is in the liver. We have a lot of different kind of cells. They can produce different kind of enzymes for digestion and for metabolic pathways, like stage 1, stage 2 metabolism, right? So we have neurotransmitter, hormones, right? Hormones is also specialized cell, like for instance, thyroid, right? Thyroid hormones produce it in your thyroid. So all those cells, they are triggered, okay? They're triggered to produce these hormones, okay? And then after that, the hormone itself is a trigger receptor, okay? They trigger other parts of the body. And we have channels and pores. So let's say your cell have certain injuries, right? Let's say DNA injuries. What can happen is that there are 3 possible pathways that can occur. One is that the cell dies, usually through apoptosis, which means that the cell will, the DNA or the cell, certain chemicals in the cell will give signals, okay, signals saying that this cell is injured. And then the cell will start to produce a lot of oxygen radicals, reactive oxygen radicals, or what I call as ROS. And then you will start to attack all over the cell, and you'll see that the cell will die. Sometimes cell injury is not critical. Okay? If it's not serious, you have the cell repairing itself. Right? So repairing itself means that sometimes you have repair enzyme. Okay? Those repair enzymes can actually go into the nucleus and starts to repair the DNA. Right? So in that case, your DNA can be restored, and then it becomes back to the healthy cell. Another one will be the DNA changes. And this is the one that is undesirable, right? This can actually happen very frequently. In fact, every day we have a lot of different cells in our body having DNA change, okay, due to the complex reaction that involve DNA replication, right? So because there are so many steps, right? So definitely every day you will see a lot of different changes to the DNA, right? But most of the time they don't lead to cancer cell. Okay, the changes usually they are destroyed by the body itself, right? Because we have evolved over millions of years, right? So our body is capable of releasing, okay, destroying all those cells that are not really useful to us, and it has changed, right? So it's those that are not destroyed by our body and starts to replicate. That's the one that is something that we should be concerned of, because these are the one that leads to cancer cell. Okay. Okay, replication, just to go a bit into details, right? So DNA replication usually occur, as I mentioned, through the action of multiple enzymes, right? Usually DNA polymerase, which will go into the DNA and starts to divide. Okay, there's a helix unravel. Okay? If you look at DNA structure, it is double helix, right? So the helix will start to unravel, okay? And then the strands part. Okay, the strands will part, and the DNA will start to replicate based on the enzyme reaction in the DNA polymerase. Right. So this is what we call as the mitosis and meiosis process. Right. Mitosis is when you have a somatic cell division. Meiosis is when you have like your sperm. The sperm is dividing. The cell is dividing to become sperm. Right. So the number of chromosome, right, for mitosis is, let's say if you are human, right, 46, and then meiosis will be 23. Okay, half of that. Right. So not always perfect process. That's why we need a certain enzyme to help us to repair if there's a problem. Then this kind of enzyme, we call it as the repair enzyme. Right? So the function of replication will be to duplicate the DNA. Right? Replicate means that we want to have the same type of cell, right? So that they can actually have the same information, right? So mitosis, as I said, somatic cell with the 2N chromosome, right? Whereas your meiosis product will be your N, okay? 23 for human, right? So usually for this process, right, mitosis, number of quality and quantity of chromosome, they are preserved, and the trigger for mitosis to happen, okay, will be these external signals, okay? Signals from your body to say that your this part of body is injured, then your cell starts to divide, okay, to repair itself. Then we have hormone. Okay, hormone can trigger mitosis. Okay, like growth hormones, right? Many growth hormones can trigger this mitosis so that we can grow up. Okay, you can see in the children. Children, they have a lot of this growth hormone, right? And internal factors inside your body. Okay, and then growth factors. Right? So sometimes if you eat certain medicine, it goes in your body. It can also produce certain effects that promotes mitosis. Right. So this is how it looks like. So you start off with the cell, and then after that you have the different phases like prophase, metaphase, anaphase, and through this process, right, in the end you end up with two different daughter cells that has the same genetic information, right? compared to the first cell. Okay, so if you look at meiosis, meiosis is another process, okay, whereby this is concerning the germ cells that divides into gametes. Right? So that you have two cell division which produce 4 different daughter ions, daughter nuclei, right? So this is mostly in our reproductive cells, right? So what happens is that this is 23, okay? Let's say this is, the other one is 23. This is male and female, right? So both will be combined later on, form 46, right? Okay, so once DNA is replicated, right? So then it will become 2 copies of itself, right? So the DNA itself is useless if it cannot be used to send out signals, right? So we have something called the RNA, right? RNA is ribonucleic acid. This RNA can copy the information in DNA, okay? And then after that, it moves out to the cytoplasm, right? And then it starts to carry out certain processes, okay? With the aid of certain proteins like ribosome to produce what you call as proteins like enzyme. You have like hormones and so on, right? So you can see here, you have the DNA, right? So this happens in the nucleus, right? So DNA cannot move out from the nucleus, right? So it needs RNA to come in and transcript all the information, right? And then after that, it moves out, okay, into the cytoplasm and starts to have the ability to form this polypeptide, right? Polypeptide like protein, okay? And then it comes with the help of ribosome, right? So in this area of chemistry, right, is very hot. I would say hot, because if you look at the Nobel Prize, right, every year, they award Nobel Prize in the biochemistry. Of course, this year is a bit different, but it's quite related to biochemistry also. This year's Nobel Prize is by, I think it's click chemistry, and because the resemblance, as in the use of click chemistry can be applied in biochemistry, right? So most of the Nobel Prize in chemistry nowadays looks into this kind of things, like how you can investigate certain processes in the cell and so on. So ribosome is actually one of the area whereby there are several Nobel laureates, right? For instance, if you look at the structure, okay, structure of ribosome, it was solved by several people, Professor Yoda, Professor Vanke, and another one, I can't remember his name, but then they are the ones who started this ribosome structure solving. So actually, I did meet one of them, Professor Vanke, in the Nobel laureate meeting just early this year, and he shared with us several things. Okay, what leads to his discovery of the structure of ribosome, right? He mentioned that he was in the field of physics, crystallography, and then he tried to look into like research in crystallography, and then he found that the biology part is the one that is hot, the one that is interesting. So he eventually moved into the biology field, and then he take in his crystallographic skill into that area, right? So after that, he combined, in other words, he combined crystallography skills with the biology area to solve the structure of ribosome, right? Eventually, he managed to get it. He get the structure based on his physics skill, and then he opened up a new research area, right, in this ribosome chemistry. Yeah, so eventually he got a Nobel Prize for that, right? So as I said, this is quite a very hot area for people to explore. If you are interested to get Nobel Prize, right, because go into biochemistry. Right? This is just my suggestion. Okay, so DNA translation occurs in the cytosol. Okay, cytosol is the part outside the nucleus, right? So you have the RNA going out from the nucleus, and then after that, it starts to translate. It starts to build proteins, right? So you have different kind of RNA, like mRNA, tRNA, like messenger RNA, transfer RNA, right? So all these, they are the one that is going to work to produce the protein that you want, right? So what they did was they go into the cytosol, and then after that they combine with certain proteins like ribosome, and then after that they start to form amino acids, right? And then these amino acids will then combine with each other to produce polypeptides, right? Okay, just to brief you a bit on the structure of DNA. So DNA is actually double strand of nucleotides. Nucleotides, they are nucleic acids. These are the different kind of nucleic acids that you can have: cytosine, thymine, uracil, adenine, guanine. Right. So they are actually arranged in a sequence, right? Sequence. And then if you look at a lot of times people say we're doing gene sequencing, right? And they're talking about how they look into this kind of sequence, right? The sequence of the amino acids in the the sequence of your nucleic acids in your DNA. So they are actually looking into this. Okay, they manage to sequence the gene, right? But sequencing gene does not equal to knowing what their function is, right? So even though they know the gene, and they don't know what the function is, that requires additional investigation to look into how a gene can be expressed by our cell. Right. So another thing is codon. So codon is a sequence of 3 bases. Right. So this is a definition. Just to show you how the this RNA can be translated. Just a minute. Yeah, I just want to show you this video. Okay. Okay.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional protein. Into the amino action, use the
just to show you this chain here, the yellow color one is actually your RNA, right? So this is your ribosome, right? The bluish color one, and then as this RNA is going through this ribosome, right, then you will see that this red color thing starts to come up. So this is your protein. So when your RNA is doing translation, right? So your protein will be the end product. It will come out from the ribosome, and then after that, it will be built into maybe an enzyme or sometimes hormones, depending on what kind of information your RNA have, right? So you will see that a lot of this protein will come and go and come and go. And during this phase, you have translation happening, right?
The mRNA.
Okay, so another one is transcription. It means copying. So we are copying the DNA information so that we can send it out from the nucleus. Translation, it means protein formation, right? You have different kind of RNA, mRNA, rRNA, and tRNA. So mRNA messenger, R is ribosomal, T is transfer. Okay, just more information on the DNA structure. You have nucleotides form chain. Okay, this is the typical DNA structure which you can see that there are a lot of different kind of nucleic acid like guanine, cytosine, thymine, and adenine. For RNA, of course, if you look at the structure, RNA don't have this thymine. Instead, you have uracil, right? So that's the difference between DNA and RNA, right? So multiple codons forms gene, multiple genes form chromosome, and multiple chromosome form your DNA, right? So sometimes there can be certain errors, right? Error means that your DNA have problem, whether it's in terms of during replication, you have defects, or sometimes it's due to chemicals, right? Chemicals, toxicant, like for example, aflatoxin, which can go into your nucleus and bind with certain nucleic acid. Okay? That will cause a lot of issues in terms of the translation later on. Right? So you can have error during base pairing. You can have error due to the repair enzyme inability to repair your DNA. Right? So you also inside your cell you have regulatory genes, operons, termination sequence. So if during replication there's no termination, then your production of DNA will be continuous, and then after that you will see that you will end up getting defects. Okay? Sometimes it can lead to excessive growth of the cell. Then you have methylation, okay, which means addition of methyl group into certain part of the DNA. And you can also have issues during post-transcription and translation processing. Right? Just to go through some brief chemical interaction that can cause your DNA to be affected. Okay? So there are many kind of toxicants, okay, that can affect the quality of the DNA. Right? They can cause chemical reaction in your DNA. If there's a defect, right? Let's say this is your DNA strand, right? If the mRNA comes in and then it is transcripted, maybe you can have the same thing, right? So if there's a certain defects, for instance, here you have an additional CH3 group, what will happen is that your RNA will be maybe something that is different, right? So this RNA, because of the structural difference, when it goes to the ribosome, right? Let's say this is your ribosome, and then this RNA, right, goes inside your ribosome, and then because of the structural difference, the protein that comes out from the ribosome will be something that is different from what is intended. So this protein, let's say it's enzyme, may not be able to function as what it is intended to do, right? So for this case, that will mean that the cell has to die, right? The body will have send signal to the cell and then say that this cell has defective site, and then after that you have apoptosis, right? The cell starts to break down, and that's it. Okay, it's gone. Okay. But of course, if you have certain more serious effects, for instance, your DNA is defective, and then it starts to replicate excessively, right? So that will lead to cancer, right? So there are many kind of chemical interaction. For instance, you have alkylation. Right? Alkylation means that addition of certain chemicals through covalent bond. Right? For example, here if you look at this, right? If you have alkylation, you may have certain chemicals coming here forming, right? Or sometimes can have attachment of the benzene ring or some chemicals to the strand. So you have intercalation, non-covalent binding of chemicals between two adjacent base pair, right? So you can also have cross-linkage. Okay? Cross-linkage means that inter-strand covalent binding. If you look at if you look at this diagram over here, intercalation is something like this. You have the binding here. Okay? So this is a double helix strand whereby you have additional binding, covalent binding due to intercalation, whereas this one is cross-linkage, okay? Binding of this covalent bond interchange, inter-strand, right? And then you can also have another one is breakage. Okay? Breakage means scission of one or both strands of DNA. Usually breakage is due to this ROS, reactive oxygen species, right? So if you take in certain food, right, that can lead to the production of ROS in your body, right? For instance, PAH, right? Right. So it goes to your body, it starts to produce a lot of radicals, and these radicals can damage the DNA, right? It can attack certain parts, okay? That leads to the scission, okay? Because the ROS is very, very reactive, right? Okay, so this is aflatoxin. This is one of the chemical found in fungi, right? Fungi, you can have it in the corn, right? For example, the corn that's been produced, if it's not taken care of properly, right? So all these fungus can grow on the surface of the corn and starts to produce this chemical called aflatoxin, right? So this aflatoxin is considered as a genotoxin, right? Genotoxin because it can bind with your DNA, okay, through this this technique, alkylation, right? So it can cause alkylation of a DNA to have the additional group. For instance, here you can see the guanine group is bounded with the aflatoxin, right? So this affects the translation later on. So we have gone through this, how a DNA can actually be replicated, and it can actually transfer information from the DNA to the production of protein, okay, through transcription and translation. So if you look at this kind of process, right, there are many different kind of enzymes, there are many different chemicals that are involved in this process. So if that's the case, if certain toxicant can affect a specific process, it will cause toxicity, right? So just to give you some introduction of all these terms, right? Okay, we have genotoxic. Okay, so the genotoxic means that that particular toxicant can affect the DNA, right? It can alter the DNA to some extent. Okay, so you have to remember that altering the DNA itself is a very general thing, right? You can alter it, and then after that, that cell is destroyed by the body. Okay? It does not lead to anything. Okay? Nothing happens. The DNA got damaged, and then the cell destroyed, just like that. Right? Of course, sometimes your genotoxin can actually affect the DNA to an extent whereby it can cause mutation of your cell. Okay, for that case, we can call that as the mutagen too, right? In other words, if you look at genotoxic, right, it's actually a very broad field, right? Genotoxic. And below this genotoxic, you have mutagen. So mutagen is part of the genotoxic class, right? And then we have teratogenesis. Teratogenesis means that that particular toxicant can actually affect the fetus, right? For instance, when you eat something, a chemical, right, into your body, let's say the person is pregnant, these chemicals can move through the placental barrier and then goes to the fetus and cause the fetus to have certain defects. That kind of toxicant, we call it as teratogen, right? So that's the difference between teratogen and mutagen. Teratogen is concerning the fetus itself, okay, the baby in the body. Okay? Whereas mutagen, we are talking about the ability of the genotoxin to cause certain damage to the DNA that resulted in mutation, right? On the other hand, if you have carcinogen, right, carcinogen, we are talking about the genotoxin that can go into the body, go into the cell, go into DNA, and then cause cancer effect. Okay? So not all carcinogens are genotoxin. Okay? Some carcinogens, they are not genotoxin. Some carcinogens, they don't affect the DNA. They affect other things. Like for instance, they affect the ability of the cell to produce certain hormones. They affect the certain receptors and so on. Right. So these are the differences. Right. Genotoxin is a large class of chemicals that sometimes have mutagenic effects, sometimes have carcinogenic effects. Right. So genotoxin are the chemicals that can bring changes to the DNA and RNA. Right. It only brings changes. Right. And then teratogenesis is when the chemicals can affect fetus. Mutagenesis is when the mutagen can affect the DNA and cause mutation. Whether it's good mutation or bad mutation, it doesn't matter. As long as there's a mutation, that is what we call as a mutagen. And then carcinogenesis is concerning carcinogens that have the ability to cause cancer, right? The end point is cancer, right? We'll look into one by one. What are the difference between genotoxin? What are the difference between carcinogen? What are the difference between all these different terms, right? So genotoxicity is concerning, as I said, the ability of a certain chemicals to bring changes, right, to the cell genetic materials, right, DNA, RNA, right, and then affects the integrity. Right? So every single day we have DNA damage, right? DNA damage in our body. That's very common, right? 10,000 to 1 million times per cell per day, right? We have DNA damage, right? So most of the time, the DNA damage will not result in anything that is problematic. In other words, our human body, they can repair this DNA damage, right? So the alteration may be permanent, may not be permanent. So when you talk about the chemicals go into the cell and then it starts to cause damage to the cell. And this is very natural, right? There's nothing unusual about it. Okay? Most of the time, the DNA damage is not transmittable. And this is like 95% to 99% is not really a problem, okay? Non-transmittable, which means that there's no mutation, there's no significance, there's nothing happened, right? It is just like damage and we destroy it instantly, right? But sometimes it can have transmittable DNA damage, okay? Which means that that particular damage leads to a mutation, right? So this is the mutagen, right? Genotoxin can be mutagen, right? So when you have transmittable DNA damage, which means the replication of the cell itself starts to produce the same DNA that is damaged, right? So it starts to replicate, and then you have a lot of damaged DNA, okay? Then that is when the mutation will start to happen, okay? You may end up getting side effects like cancer or maybe some good mutation. Okay, for instance, you can have more resistance to a certain bacteria or maybe infection. Okay, the sources of genotoxic agent, they can be from environment. For instance, river, lake, pond water, air pollution, right? So if you walk out from the campus, maybe in USM, right, you will see that there's a big lake, a giant lake over there, and in the lake itself, you have a lot of chemicals inside. Okay? You can look at the color of the lake, right? It's greenish in color because it's polluted, right? So this kind of lake sometimes can contain certain chemicals, right? Or sometimes the green color, right? It is actually algae, and they produce certain side product due to their respiratory needs, right? So they can produce certain chemicals. All these chemicals, they can be considered as toxin, right? Genotoxin, which can affect the DNA, right? So we have also drugs like chemotherapy drugs. Chemotherapy drugs, they actually can be very useful for cancer patient. Okay, but then sometimes the drug that you're taking may have certain side effects. Okay, that leads to something called a secondary cancer, right? And then you have antibiotics. Antibiotics, as in it is generally just a minute. I have a class. Yeah, so sorry. You have drugs. Okay, drugs like antibiotics. Okay? Antibiotics is something that we feel like is good, right? We sometimes eat it during our time when we are sick, right? In fact, I have certain relative, close family member who take in antibiotics during pregnancy, right? So we thought that the antibiotics is something that is useful for our body, but then it is not. Right? Sometimes it can cause certain effects. So this close relative, she ate in these antibiotics into the body, and then after that, it was during the early stage of the pregnancy, about 2 weeks, 2 to 3 weeks of the pregnancy. She consumed the antibiotics in the body, and then after a few weeks, she went to check the baby, right, the fetus, and eventually the doctor said that there's no fetus, right? And then Of course, there's no link. The doctor did not say that it's because of the antibiotics that caused this to happen. But then we actually suspected that the antibiotic itself is the one that leads to these effects, right? Leads to the body started to have certain effects to the embryo or the baby or the fetus, right? So aside from that, you have the substance abuse like heroin, LSD, cocaine, and morphine, which are the sources of genotoxin agents. Chemicals like pesticides, industrial waste, formaldehyde. Formaldehyde, you can find it in furnitures, right? Sometimes they use formaldehyde to bind certain things in the woods, right? That will be released, right? If you are not careful, right? So it can be released to the room, and then because the room is enclosed, right? So it can be absorbed by human. Then we have radiation, like sunlight, radon, medical radiation therapy, which are some of the sources of genotoxin. Yeah, so some example, aflatoxin. For instance, you look at this corn, right? So those corn that we purchase from the supermarket, they are quite clean. I would say that they are okay to consume. But sometimes those contaminated one, right? The contaminated corn that has this fungus growing, right? They may contain aflatoxin, right? So this aflatoxin, as I show you just now, can affect the DNA, and then the DNA can actually be alkylated, okay, which leads to error in translation. Aside from that, you have radon gas. Okay, radon gas, which can be found in the your light bulb, right? Sometimes you have uranium in the environment. You are not aware of the uranium, right? Which can indirectly go to your house. Right? This is quite common in certain areas, whereby you have uranium in the environment, but they are not aware of it. In Southeast Asia, I think it's quite safe. But then in countries like in Russia, in China, specific part contains uranium, which is quite dangerous, can be a source of genotoxin. And then products like furniture sometimes utilize formaldehyde, right? This is quite a dangerous chemical, right? So I have a project working with formaldehyde. Right? We are actually trying to find a solution, okay, to remove this formaldehyde. Generally, if you look at those like hospital, right, they want to preserve organs, right? They also use formaldehyde, right, as one of the preserving agent. So I have one project that looks into how we can actually neutralize this formaldehyde, right? So the formaldehyde itself is quite toxic, okay, because once you absorb it into the body, right, it can go to your cell, it can cause DNA damage. So this is something that is undesirable, because once you have DNA damage, it will certainly leads to cancer, right? And this formaldehyde is actually a confirmed, okay, carcinogen, right? So I have a student who is pregnant, who wants to work in this project, but then I have to reject. Okay? I have to explain to the students this project is about formaldehyde, and then I have to explain that this formaldehyde is a carcinogen, and also to some extent it is also teratogen, right? It can affect the fetus. So when you want to work with something that contains formaldehyde, right? So just be careful, right? Be very careful. I have a big glove box for this project. Okay, so you have to use a glove box and a lot of protection, okay, to avoid this formaldehyde from getting into your skin, right? So today, if you are exposed to formaldehyde, you may not see any problem, but after a certain time, right, you may start to notice that there's certain changes. It may take like 2-3 years for formaldehyde to express itself, okay, as a cancer, right? So the other one is the BPA. Okay? BPA is bisphenol A. This is also one of the endocrine disruptor that can cause mutagenic effects to the body, right? So it can also mimic mimic endocrine. It is an endocrine disruptor. It can mimic the estrogen, right? Which means that once you take in BPA in your body, you start to have some effects. For instance, feminization. If fish take it, they will become like having several reproductive organs, right? Like female and male reproductive organ together, right? This is also a problem today. Just want to share with you another one common source of this genotoxin is sewage sludge. Right, sewage sludge is something that is produced in a wastewater treatment plant. Wastewater treatment plant is a place whereby most of the time use microorganisms, right, to remove pollutants. So this microorganism will consume a lot of those waste that we throw into them, and then when they consume, they start to reproduce, right? So they will start to reproduce into this what we call as a sewage sludge. Okay, you can see this brownish in color, right? So there are many areas whereby they take this sewage sludge, they dewater it, okay, which means they remove all the water, and it becomes this kind of color, blackish in color, right? So the sewage sludge is rich in nitrogen, and they are to some extent rich in phosphorus and carbon, right? Because they are proteins, right? They consist of proteins. That's why they have nitrogen. Certain parts contains phosphorus, like their DNA contains phosphorus, right? They themselves, the sludge is made out of carbon, right? So many areas they like to use this sludge and they apply it in landfill application, like agriculture. Okay, they use it to grow a lot of plants, right? I know in certain part of Malaysia they actually did this, right? In Indonesia, I also, if not mistaken, I've heard that some people do this, right? But it is actually not safe, if you have this kind of process, because most of the treated sludge, they are contaminated with certain chemicals. For instance, you have heavy metals, you have hormones, you have all those microplastics, right? So all these chemicals can actually be absorbed by the plant. Okay? And then because of this, if you consume this plant, right, you're actually consuming all those chemicals, heavy metals and so on, right? Okay, the good thing about this sludge is that it has macronutrients, soil enzyme, high organic matter. But the bad thing is that it consists of a lot of genotoxin, right? Like pathogens, microplastics, pharmaceutical active substance. This is a genotoxin. This is also genotoxin, and this is also genotoxin, right? So a lot of these chemicals in the sludge can actually affect the DNA. But the other one is the pesticide exposure. So pesticide exposure generally like glyphosate. Right, when you're exposed to glyphosate, it is to some extent not so toxic, but then if the exposure is at a high dose, then of course it can cause certain effects to the health. It can alter the DNA because it's considered as a genotoxin, right? So it can also bind with certain heavy metals, and then the toxicity of the glyphosate changes, right? It becomes something that is more or less toxic, depending on what kind of heavy metal it's bounded with. So yeah, all these are the example of genotoxin, UV light, tobacco, dioxin, asbestos, ammonia, and sometimes air pollution, processed meat. Even processed meat is considered as genotoxin because they use benzoic acid as a preservative. Sometimes they use certain chemicals that, even though it's allowed, right, by the government, but they are actually genotoxin. They can affect our DNA, okay, depending on how much you eat. That's why there's a saying that we shouldn't eat a lot of processed meat. This is the reason, because it is capable of inducing cancer, right? Although it's not 100% of people who eat processed meat will get cancer, but then there's a percentage. Okay? Even the textile dye also is considered as genotoxin, right? So when you want to buy a clothes, right, a t-shirt or anything that is with textile dye, right? The color in the clothes, right? Try not to get it from those that is like the cheap one, okay, the one that is not properly recognized, right, or not properly investigated, right? So some people they open up the factory and then they use dye, low quality dye, right? Most of these kind of dye they are quite dangerous because they contain zero toxin, okay, or they themselves are zero toxin. Radiation, okay, radiation itself is also regarded as one of the zero toxin agent because they can induce changes, right? Radiation like alpha radiation, gamma radiation, X-ray, even X-ray is also something that can damage DNA, right? So that's why people who work in this X-ray area, they have to be very, very careful. Okay? Usually they have rotation, right? They cannot have a person doing X-ray for the patient every day, the same person, right? They have to have a rotation. They have to monitor their exposure. Yeah, even the one in the university whereby you are dealing with XRD, right? X-ray diffraction technique, right? Certain university they will need to undergo training and to get license, right? In Malaysia, we don't have this kind of license. I believe most of the operator of XRD they just do it, right? And then they don't really take the proper precaution because there's no clear guideline. But in countries like in Singapore, they actually have this training. Okay? They need to undergo a training. They need to take exam, and then they have to have the license to run the XRD. Okay? Even though the machine is very small, or maybe just like for maybe education purpose, they also need to have a license as long as it's related to this X-ray, right? So you have to be very careful when dealing with radiation. Even UV radiation, you should not be exposed to it that often, right? So when you have radiation, you have damage to your DNA. So we don't know what's the extent of damage, right? Sometimes the damage can be so severe that it causes cancer, right? Oops. Yeah. Example, look at the bird here. You can see, yeah, there's a tumor growing in the face of the bird. Beside its beak, and then you have this this I think it's a mouse, right? You look at the eye; it has a certain tumor growing. Okay? These are all due to the exposure to genotoxin, right? Okay, then we have something called a teratogen, which can affect the fetus, right? So if you consume teratogen to your body, the mother may not have any issue, right? Sometimes the mother has certain resistance to the chemical, but then the fetus may not, right? So if you look at the slides that I shared to you in the first lecture, we talked about thalidomide, right? So that was the chemical that is introduced the population, and then there's no proper study and proper toxicology study to look into the effect of the thalidomide to the population. So no one knows that it is a teratogen until a lot of cases whereby the baby born with a lot of congenital malformation, right? So this is a very crucial what you call areas. Okay, a lot of studies have been conducted to look into what kind of tetragen important and how they can actually cause problem to the embryo and the fetus, right? It's a very complex mechanism, right? So most of the time, when you're exposed to tetragen, your body knows, and then it will start to expel the embryo and fetus through something what we call as a natural spontaneous abortion, right? This is natural, right? The chemical that a lot of people eat, okay, eat to abort the baby. I don't know whether you know about this chemical, some drugs. I think that there's some drugs or some medicine whereby if the mother feels like you don't want to have a baby, okay, the doctor will prescribe this drugs to the person, and then the person eat it, and then she will have spontaneous abortion, right? So the doctor actually give tetratogen to that person, right, to have this kind of spontaneous abortion. And usually this will work only in the first to 8 weeks of the gestation, okay? Which means within 2 months of pregnancy, right? But of course, sometimes exposure to tetrogen is not always end up with a spontaneous abortion. Sometimes you can have the fetus survive, right? Survive through the process, and then when the fetus is born as a baby, you will see that there's morphological defects in certain tissues, right? And then because of this, then of course there's a term. Okay, some people call this as a monster, right? Which is very sad. Example, okay, example is like if you can see, sometimes these frogs can be born with 5 different legs, right? This is probably because of the tetratogen, or maybe sometimes it's because of the virus. Right? There are many reasons, right, why these kind of things can happen. This is a classical example of exposure to tetratogen. This is what we call as a cyclops, right? Cyclops effect, right, which comes from the exposure to certain tetratogen. Sometimes it's because of the herbicides, right? You can see that this is I think it's a goat, okay, with only one eye, right? So this is things that can happen when it's exposed to tetratogen, right? So I actually want to show you this video whereby there's a lady. This is a lady, right? A lady found. I'll just explain to you. A lady found a frog with 5 legs, right? So she is very excited with this discovery. Okay. Some people, they have no exposure to this area of research, or this area of science. They don't know what is happening, and when they saw 5-legged frogs, they feel like, oh, this is a wonder of nature. Sometimes they saw two-head snake. They feel like this is from nature and so on, right? It's from God and everything, but it's actually not. It's actually because of human, right? So we introduce a lot of chemicals into the environment, and then all these animals starts to have these effects. They start to suffer from the effects of the chemicals that we introduce. It alters their gene, right? And their offspring have a lot of problems. For instance, this 5-legged frog, they themselves also have certain cancer, right? That's something that is very sad to say. And we will see these kind of cases happening more and more often in the future because of our introduction of different kind of chemicals every single year, right? Oops, sorry. One more study would be this. This is one of the study has been published, I think, in the literature. This is the lupin alkaloid birth defects, whereby a baby boy was born, right? Born in the US, right? With severe deformities in the bones, arms and hand, and also certain parts. They are absent, partially absent. Like for example, these forearm bones, right? So a baby was born of these defects, and then after that, the doctor starts to investigate. Okay, this kind of effects can be due to many reasons, like genetics, and sometimes it's due to environment, right? So the doctor did some extensive investigation and found that genetic is not the factor, right? So they found that it was the environment itself that caused the baby to have these kind of defects. So they starts to investigate, and then they found that the population at that particular area likes to drink milk. Okay? A lot of different milks, in particular goat milk. Okay? So they like to drink goat milk, and then they also found out that this area, right, the goat has been exposed to herbicides, okay, or certain pesticides, okay, which can have a certain teratogenesis effects, right? So because the population drinks this goat milk, right, they are actually affected, right? So this mother actually drank regularly throughout her pregnancy, every single day, and I'm not sure whether she did ultrasound during pregnancy, but most likely no, I don't think so. And then once the baby was born, then of course, that's the point where they found out that the baby has a lot of deformities, right? So this is a sad case of exposure to teratogen, right? Okay, then we have mutagenesis, whereby we'll talk about the effects of mutagens, right? This is a component of genotoxicity. Like as I said, if this is a genotoxicity, you have a small portion of it as a mutagen, right? This small group, small subset of the genotoxicity, right? Generally, it means that the genotoxin induce an effect to the DNA until it can cause a mutation, right? So this kind of mutation can be good, can be bad. Okay? It can be beneficial in the sense that it helps us, okay? Or it can lead to cancer. If it leads to cancer, that means the mutagen is actually a carcinogen, right? If it doesn't lead to cancer, then we can just call it as mutagen, right? So the beneficial effects, like it can give you HIV resistance, it can give you malaria resistance, and then those antibiotic resistance in bacteria is also due to the mutation effect, right? But of course, the bad thing, sometimes the bad effect is like a cancer, you have sickle cell, you have all those diseases. If we talk about mutation, we're talking about somatic cell mutation, which will lead to metabolic dysfunction and cancer. If we talk about mutation in a germ cell, like sperm and ovum, it will lead to heritable change, right? We also have several kinds of mutation. One is point mutation, whereby you have substitution, you have frame shift, okay? That means the shifting of the base, or in other words, the nucleic acid can actually shift. And when you have shifting, your translation will lead to error in the production of the protein. Then you have chromosomal aberration, okay, which leads to the structural anomaly. Okay? The structure of chromosome is different. Or you have numerical anomaly, which means that your chromosome may have additional chromosome, right? For instance, Down syndrome, you have 47 chromosomes, whereas a normal human, you have 46. Right? These are the type of mutation, right? And you have the original one. Okay? You can have base substitution. You can see, right? This T is substituted by C, right? Thymine is substituted by this C, right? You have base addition, which means the addition of another one species. Okay? You compare this and this, you can see that the base addition is actually longer. Okay? You have base deletion, which means one of the base is removed from the DNA. Okay, so the other type would be this type, okay, deletion, okay, whereby certain part of the DNA is deleted. Sometimes you have duplication, okay, whereby you can see this part is duplicated. You have two different similar parts. The other one is inversion, right? The same part is inverted around. It is changed around. So this kind of effect usually is due to the enzyme error, right? It can be due to enzyme error, and sometimes it's triggered by what we call as the mutagen, right? Mutagen goes inside, and then it can trigger this cell division, mitosis and meiosis, and it affects the well-being of the chromosome, right? So if you have Down syndrome, of course, you have additional chromosome, right? So this is what kind of karyotypes, which helps us to examine your chromosome, see whether it is 46, okay, or it is not, right? So if you look at this, right, you can see that you have X, you have Y. So this is a male. If you have both X, that's a female, right? Yeah, this is a chromosome aberration due to defects in the chromosome. Sometimes you can have loss or gain of the complete chromosome, which means, let's say for instance, Down syndrome, you have extra 47, extra 1, which makes it 47, And of course, you can Google the image Down syndrome. You see that an additional chromosome to human can actually lead to various effects. Okay? The characteristic of Down syndrome patient is very different from normal human. Okay? The other one is Turner syndrome when you have 45. Okay? That means you have absence of one chromosome, right? And that will lead also to certain effects, which can be translated to various symptoms. Okay, for instance, there's underdeveloped reproductive organ, that person have shorter fingers and so on. Right. The last part is this carcinogenesis, right? This carcinogenesis is what we call as a mutation that can lead to the production of normal cells being transformed to cancer cell, right? Sometimes you call this as oncogenesis, right? You go to hospital department, when you want to go to the cancer department, right? They don't call it as carcinogenesis or so on. They call it as oncology, right? That's the cancer department, right? This is a very, very complex process, right? Carcinogen can be genotoxic or non-genotoxic, which means that a carcinogen can actually either affects the DNA or it does not affect the DNA, right? So there are two different groups of carcinogen, right? If it is genotoxic, that means the cancer is due to the interaction of the carcinogen with the DNA itself. But if it is non-genotoxic, it means that that particular toxicant does not directly interact with DNA. It can interact with hormones, it can interact with organic compounds, or it itself is a hormone. It can induce a lot of different effects, but it does not interact with the DNA directly, right? That's the non-genotoxic carcinogen. Okay, we have three categories. The chemical carcinogen. Okay. Those are from those things like aflatoxin, benzene, asbestos, right? And then we have physical carcinogen, like UV rays, X-ray, gamma rays, right? All those high energy radiation, which can induce change in your DNA. And then you have oncogenic viruses, which means cancer-causing virus, like HPV, right? This HPV is very, very problematic, right? There are many campaigns in Malaysia, right, that promotes taking vaccine for HPV virus. Okay? So one of the reasons is because HPV can cause cervical cancer, right? It is capable of inducing growth of tumor in the cervix, right? For female, because for male, of course, it does not cause cervical cancer because male don't have any cervix, right? But it is also a problem because male can actually transmit HPV at a much greater percentage to female, right? You also have this hepatitis virus and all these viruses. They are considered as oncogenic viruses, which can cause cancer. Right, two different ways, as I mentioned. One is it can directly damage DNA. The other one is it can cause damage and inflammation, result in cell division. Right? Okay, so carcinogenesis is a very complicated process, right? To get cancer is very, very complicated. It has, but it can happen to everyone. Okay, even though a person is healthy and is not exposed to environmental pollution, it can still happen, right? Because of the complex replication of the DNA, right? So if you look at this figure, right, we have procarcinogen, and this procarcinogen means that it is a chemical, okay, a chemical that is not carcinogen but can be triggered to become a carcinogen, right? So for instance, like benzene, when you talk about benzene, if we consume benzene itself, it is regarded as not carcinogenic. But if it goes to your liver and then it starts to be metabolized via stage 1, stage 2 metabolism, you start to see that this benzene is transformed into epoxide, which is carcinogenic, right? So this is what we classify as pro-carcinogen. Sometimes it can start off directly with carcinogen. So when you have this kind of chemicals, they can actually interact with your DNA, right? So if it's severe enough, of course, it will cause mutation to the DNA, and then this mutation directly cause cancer, right? But often this is not the case. Most of the time when we consume foods, consume this benzene, PAH, and all those chemicals, you will start to have mutation, okay? Or it will be in the initiation phase, right? So this initiation phase is when the cell itself starts to have damage to the DNA. Okay? And this damage to the DNA can stay, and the cell can stay in your body for many, many days or sometimes months or even years, right? Without anything happening, right? So, but then there's an initiation, which means that your cancer cell has initiated, right? This initiated cell can actually be destroyed by your body itself, right? It can be destroyed by your immune system. It can be destroyed by signals that send to the cell to self-destruct, right? Okay? Nothing will happen, but it is when you have the promoting agent, right, that comes in, and this promoting agent will promote the initiated cell to divide excessively, and then after that you have the progression stage, whereby it starts to progress, it starts to metastasize, and it will lead to cancer, right? So it is actually a very complicated process that has a lot of different steps, right? So just to share with you a story, last time, this is also a very, very famous area to get a Nobel Prize. Last time I met a Nobel laureate who is excellent in this cancer, right? In oncology, right? I think his name is Professor William. He got his Nobel Prize in 2017 or 2019, I can't remember. But he did share with us that this kind of research is actually very, very difficult to do. Okay? It involved a lot of trial, okay? Animal trial. He also shared with us a lot of tips, right? We should how we can actually study all these kind of things, and it requires a lot of funding for us to do this kind of research. Of course, unfortunately, he lost his wife because of cancer, and this is one of the motivation for him to actually look into this area and try to find a solution for cancer, right? And I would say that this is one of the growing problem, one of the major problem in human, right? I think he is one of the person who said that the government in US and even the world, right, should promote research in looking into a solution for cancer instead of sending people to the moon again, right? He is one of the advocate for that, right? So you can look for his research, and maybe if you are interested, maybe you can join them and join this research, because this is actually a research area that can actually help help a lot of people, right? Some definition, like cancer, you can read through this. This one is in the slides that I have, that I shared with you. You can go through this. What is cancer means? It means that tumor, right? The growth of malignant tumor. Malignant here means that it can actually spread. Okay. Then we have tumor, which means that it's an uncontrolled growth of cell. Okay. And then we have neoplasia. We have benign tumor. Benign tumor is a tumor that does not metastasize. This is very common in human. For example, you have lipoma. Lipoma means a growth of fat resembling the tumor, right? Very common in many people. Right? Then you have a term called metastasis, which means that the relocation of the tumor, the spread of the tumor, right? So when your doctor, you hear a doctor say that the cancer has metastasized, that means that the tumor has already spread from one point to another point. In terms of spreading of the tumor, it depends on what kind of cancer that person have. If you have carcinoma, usually it's spread through the lymphatic system. Okay. if you have sarcoma, that means the cancer is spread through the bloodstream, right? So I also have many relatives, unfortunately, many relatives who have cancer, right? So one particular case that I remember is that one of my close relatives from my wife's side, he had bone sarcoma, right? We went to the doctor and the doctor said that this person had bone sarcoma and he has to be operated. So my relative went for operation. He was very young. He was like 17 years old, and then they detected this tumor in the bone, right? So he went for this operation and he take out this bone sarcoma, right? And then the doctor said that it has not metastasized. At that point of time, I don't know what this means, right? I don't know what all those terms means. And then I start to look into this, start to study, and then I know that the doctor is saying that he had bone cancer, and then the bone cancer actually did not spread. Okay? And then after that, they take this tumor and then they carry out biopsy, right, to see what exactly is this bone tumor. And luckily for him, okay, for the 17 years old relative, this bone tumor turns out not a sarcoma, but actually a benign tumor, right? It is not harmful, right? So this is one of the cases where he got lucky, right? So you'll see all these terms if you're studying cancer, like sarcoma, carcinoma, and so on, right? Okay, so if you are exposed to a toxicant that has initiation phase, right? So that means that that particular cell is dormant. Okay? There's no outcome if it's not promoted, but there's a damage there. Okay? In fact, this initiation cells, right, it actually exists in everyone's body, right? When you are exposed to UV ray, your body will have DNA damage, right? To the extent of damage, we don't know, right? Sometimes a certain cell can have the damage that can cause cancer, but there's no outcome when it's not promoted. And in terms of the property, there's no phenotypic difference, which means that the chromosome is the same. Sometimes you can have excess or deficient certain types of enzyme. For example, you can have ATPase, glucose 6-phosphate, and Fe exclusion as well. You can have certain resistance to cytotoxic chemicals. Sometimes it can be slow growing, sometimes it can be fast growing. It depends on what kind of cancer cell that you have, right? You can have impaired cellular communication, enhanced response to growth factors, and resistance to terminal differentiation, right? So the initiated cell itself is not something that is of concern until there's a promoting agent, right? So if you have promoting agent, it starts to grow excessively, and then that's the point where you have to look into treatment, right? Okay, in terms of the characteristic of the cell, usually the promoted cell has the same characteristic as the progression cell. They have the same characteristics because they're actually growing together. You have complete loss of growth control. It is growing, very rapid phase, right? So this is one of the ways chemotherapy works, right? Your tumor grow excessively, and then you use chemotherapy to inhibit the growth and destroy those fast-growing cells, right? Sometimes there are karyotype instability, which means that your chromosome is actually dividing in a way that it is not consistent, right? You may have loss or gain of chromosomal fragment, DNA demethylation, deregulation. During this progression stage, your gene can be amplified. You have DNA damage, okay, which cannot be repaired. It can be irreversible, and progression is the same as promotion, right? Yeah, this is how it looks like. Initiation, only one cell, okay? Exposure to just some chemicals can initiate something, okay? Something can happen, and then after that you have chemical that promotes this kind of effects, okay? It starts to grow, and then you have progression, right? Okay, this one we have gone through. Okay, there are two different types of genotoxic, which means that it acts on DNA or expression of DNA. We have another one is the epigenetic, which does not act on DNA. Okay, so example of genotoxic carcinogen. For instance, you have those like all those heavy metals. They can be acting as a genotoxic carcinogen. Indirect carcinogen and direct carcinogen, you also have it. One is the one that has requiring no metabolic activation. The other one is requiring metabolic activation. This means that sometimes when you eat certain chemicals into your body, that particular chemical itself is a carcinogen. For instance, if you have things like arsenic, arsenic, it is considered as an indirect carcinogen. If you have those like PAH or benzene, benzene you eat into your body, it can metabolize through stage 1, stage 2 reaction. It is considered as metabolic activation. But if you have those that requires no metabolic activation, for instance, formaldehyde, you eat into your body, it starts to affect your DNA directly, right? So example of epigenetic carcinogen, for instance, nitrotriacetic acid, NTA. This one we looked into it last week on how this can actually increase the solubility of lead in water, right? This is a chemical that was used many years ago. I think it's washing agent or maybe soap or washing powder, right? But then because of its widespread pollution and it caused a lot of issues in the environment because it's very recalcitrant. Then this chemical nitrotriacetate, it was banned by the government in many countries, right? Because of course some countries still using it, but most of the country has already banned these chemicals. Other epigenetic carcinogen includes like this one, cytopolrin A, asbestos. So this cytopolrin A is actually an immunosuppressant. When a person has undergo like an organ transplant, right? He has to be very careful because that particular organ does not belong to him, and the body can identify that as an enemy, right? Something that is foreign, right? So the body can actually create a lot of antibodies that will go and attack that organ, right? So to prevent that from happening, that person has to eat this cycloporin A chemical. Okay. Pros and cons, right? These cycloporin A's can actually inhibit the ability of the body to attack that foreign organs, but then it can also cause cancer, right? So it's very dangerous. That's why you can see heart transplant, right? The prognosis is not very good. Okay? So once a person has heart transplant, he can only live for like a few years. Okay? Majority of them live for like 5 years. Right? After 5 years, of course, you'll start to see deterioration in their health. Because of the many reasons. For instance, they are taking these drugs, and sometimes it's because of the resistance. Sometimes our body develop resistance to this kind of drugs, right? So it's no longer effective. Okay, this PAH requiring metabolic activation. So this is chemicals that you eat for barbecue, the black color region, when it starts to be metabolized to 7,8-oxide, which can damage our DNA. Okay, so other chemicals, you can go through this list. If you go through every list, right, you can see quite a number of chemicals in the environment that can cause cancer, right? So it's our body's ability to be able to segregate and destroy all this damaged DNA that prevents us from getting cancer. Okay, so in terms of initiator, sometimes pH can also be initiator. Okay. Aromatic amines, heterocyclic amines, all these they are considered as initiator, which means that they cause the first step, okay, the initiation of the damaged DNA, the initiation of the first cancer cell. Okay, and then promoting agents start to promote the cell from growing. For instance, you have BHT, Saccharin is very common used for artificial sweetener, right? So if you look at the label of the food, if you look at saccharin, then you try to avoid it because it can cause promotion of the cancer cell, right? So we shouldn't eat all these things, right? Like folic acids and even alcohol is one of the promoter, right? So in terms of classification, we can actually classify it as group A, B, C, D, right? Depending on what kind of data that you have. Those group A1 is confirmed carcinogenic because we have human data, whereas those other groups, they are not really considered as a true carcinogen because they have limited human data, sometimes only animal data, right? So causes of cancer, you have diet, tobacco, sexual behavior, alcohol. Pollution is only 2%. Okay? Environmental pollution contributes to a very low percentage, whereas it was the diet itself. Okay? Diet means that the food that you eat, 35% is excessive. Sometimes you can have secondary cancer due to the use of genotoxic anti-cancer drugs. So the best is try to avoid taking any drugs, okay, any medicine, because we don't know what it will cause, right? So even though the doctor prescribe like things like antibiotics, right? So we always have to be careful. Okay? Although we have to finish up our antibiotics, but sometimes those antibiotics may lead to cancer. We don't know that. There's a percentage of that happening, right? So we should try our best not to be sick, okay? Take care of our health, right? Some misconception. All this is to clear your misconception, right? All mutagens are genotoxic. Okay, this is true, right? Not all genotoxic substances are mutagenic. Genotoxic carcinogens are mutagens, right? And also not all carcinogens are genotoxic. So these are the statement, the true statement, right? So there are many misconceptions from people saying that genotoxic is the same as carcinogen and so on, but they are actually not the same. Okay? We classify them differently depending on what they do, right? So not all genotoxic agents can cause carcinogens, right? Okay, so this is the last part, just to share with you how we can look into the testing of whether a chemical is mutagenic or not, right? So we have something called a standard test battery, which consists of many different kind of tests, right? There are many regulatory agencies that looks into this kind of test. The test that they did is involve like an MS test, right? And then you have the in vitro and in vivo test, right? In vitro generally means an indirect test, right? Using cells, whereas in vivo means that they're testing using real animals, right? Most of the time using rats, right? Human test is very unlikely, especially for those that can cause cancer. It's not ethical and it's also not allowed. So that's why the next best thing would be to use the animal test, in vivo test, right? If in vivo test is not allowed, of course, there's in vitro test, indirectly using cells, mammalian cells, to look into whether that cell will have mutation when it's exposed to a certain chemicals, right? So to be honest, all these tests I have not done it before, hands-on, right? Usually, if you want to test out things that is related to this animal test, right? We don't really do it ourselves. Sometimes we can send it out to a company to do it, right? To do this kind of test requires a lot of ethical approval. Okay? Or in other words, we need ethics committee to look into our research, and then after that only we can do it. It's a very tedious process, especially those involving those animals' life. Right? More regulatory agencies is like EU's RL and then COM, right? So some of these are the pharmaceutical company, right? They have this standard test that you can look into to get an idea of whether these chemicals they are going to cause problem or not. So the first one I want to share with you is the micronucleus test. Okay, so this micronucleus test is to test out to see whether there's a formation of micronucleus. Right. So you have the cell, and then this cell is exposed to a certain genotoxin. Right. So when this exposure happens, then the cell itself, when it is dividing through mitosis, you will see that micronucleus will start to form. So when the micronucleus is formed, then we know that it is due to the damage of the cell. Okay? So when we detect microtoxin, for instance, you look at this is a cell. So we expose it to genotoxin. And then when it starts to divide during the anaphase stage, then all those chromosomes will go into one side. So there will be some chromosome that's lagging, okay, which means that they are slower because of the damage to the cell due to genotoxin exposure. So they will start to form this small dot here as what we call as the micronucleus. Right. So after they divide, right, they will become two daughter cells. And then if you observe it through microscope, or maybe like TEM, transmitted electron microscope, you'll see that instead of this nucleus, single nucleus, you will see a micronucleus here. Right. This is an indication that that particular chemical that you use is genotoxin. Right. So this is a TEM image. You can see this is how it looks like, a micronucleus. Very small. Right. This is during the anaphase period, the dividing period. Right. Micronucleus here. You have the nucleus here. Right. Then we have chromosomal aberration test. This one is to look into the chromosome abnormality. Okay, we want to look into the structure of a chromosome and see whether is there any abnormality in the chromosome itself after exposure to the genotoxin. So a normal one is like this. Okay, we look into the nucleus and we see that normal is like this. And if you have certain defects, okay, after exposure to the genotoxin, you will see that some of it may not be normal. This is due to end-to-end association. This is due to premature centromere division. Okay, and then the other one is due to break and deletion. Yeah, this one, you can see this one is a break. There's a break here, right? So there's different reasons why they are having a problem, right? Then we have in vitro test for genotoxicity using electrophoresis technique. When you expose the cell to the toxicant or genotoxin, then you start to carry out single cell gel electrophoresis. You will see that the normal one will look like this, right? Only one single dot because they move at the same time. But the one that has DNA damage, they will start to form something like this. This part, the tail here, those are the damaged DNA. Okay? They don't move at the same rate. That's why you can see there's a tail here. Okay, the good one is here, but then those that are affected is here. Right. So this is a damaged cell. Right. So Emmer's test. This is one of the famous tests used. Right. This is what we call as a reverse bacterial mutation assay, which means that we use a certain bacteria, right? And then this bacteria, they have certain mutation that makes them not able to grow, right? So we are actually using a bacteria that has certain defects in their DNA. Okay, so just to show you, right? Yeah. So this is the bacteria with the defects, right? And then we have two different things. One is that we use a control. The other one is the one that we add in possible mutagen, right? So these two samples will tell us whether the mutagen is actually really a mutagen or not, right? So you have the control, and the control will have no limited or no growth, okay? Because in the first place, the strain that we have is the one that is defective. It cannot grow, right? So maybe you only have one or two growth. But then once you add in mutagen to the other one, and you start to culture it, and then you will see that you have excessive growth, right? So this excessive growth is due to the change in their DNA structure, right? That means the mutagen induced changes that affect their DNA, and because of these changes, they start to grow excessively, right? So then we can say that this is actually a mutagen. right? So yeah, the other one is gene mutation test. We look into the gene and chromosomal mutation by counting the colonies, right? It's almost the same as the previous study. Yeah, the other one is in vitro cell transformation assay. We look into the cell. Normal one is like this, and then if it's transformed cell, it's like this. So this is the cell that has been exposed to the toxicant. Last one will be this one, test for carcinogenesis. This is an in vivo bioassay, which means that we are using the real animal. Okay, in this case, we use rodents, like rats, right? And then after that, you expose this rat to the carcinogen, and after 2 years, we look at the number of animal having carcinogens. And of course, it took a very long time because it takes long time exposure to be expressed out in their body. Okay, so thank you so much for today. Yeah, so this is the link that for you to download. If you can download this, it is quite big, this file. You can download it, and of course it's good. You can actually participate in next week's lecture, whereby we'll talk about how we can use this software to estimate the toxicity of certain compound. Okay. So for this software to work, right, you have to have this software and also ChemDraw. Right, you also have to have a ChemDraw software to generate the structure that you want, and from the structure you can import it into the this software. Okay. This software is called TEST, a toxicity estimation software tool. Right. So from there you can actually estimate what is the toxic effect of this chemical, okay, and compare it. Okay, so thank you, Dr. Fitri.
Thank you very much,
Thank you very much.
Dr. Wenda, for a very comprehensive lecture. So today, we got the great explanations from Dr. Wenda regarding to the genotoxicity, and then mutagenesis, carcinogenesis, and many example of that, include as well the teratogenesis. And the last part that just Dr. Wenda introduced, I guess this is the great informations, to how to say, to exercise the skill how to estimate. And this is the free software, Dr. Wenda.
Yeah, this is a free software.
All right. And for
Yeah.
drawing the chemical, maybe the student can use a kind of ChemDraw something. Oh,
Yes, for ChemDraw, yeah. I don't know whether ChemDraw is available for the students or not. I think it's also free, ChemDraw,
yeah.
if not mistaken. Yeah.
I guess the student have it, even
Oh,
though
yeah.
maybe not the how to say, the official one. Yeah.
Oh,
So
okay.
so to all students, please kindly follow this link, and then please provide it for the next week lectures. Actually, we almost approach to the end of the lectures, but just in case you have any questions, I guess we still have a few minutes for discussion. So please, any questions from student? All right. So from Dr. Wenda, I experience to how to say, to consume medicine, especially for antibiotics, and then the doctor always suggested us to fully consume, even though we already recover from our ills. So what actually the science behind, why we have to finish that dose?
Oh, yeah. So it's very important for us to
because
finish
on the
our...
other hand, I just consider if we consume over, I just worry it lead to other effects just you
Yes, but
mentioned.
for antibiotics, we have to complete it. Every time the doctor asks us to complete antibiotics, we have to finish it. There's a reason behind this, It's because of the development of antibiotic resistance bacteria. So when we consume, antibiotics into our body, let's say the doctor gave us an antibiotic dose for 7 days, right? So let's say we eat until 3 days and we feel very healthy, there's no effect anymore, and we stop taking it. At that point of time, right, some of the bacteria may still survive in our body, which may develop resistance to our antibiotic. So because of this, right, sometimes this bacteria this microorganism, pathogens, will grow excessively, and then at the point of time when you consume the same antibiotics again, it will not work anymore. This is the reason why we have a superbug problem. If you look at the US, the United States, right, there are many reports saying that all the antibiotics that they tried no longer work anymore. One of the reasons is because that person consumed antibiotic and then after that stop halfway, and then after that change antibiotics, stop halfway, and so on. So yeah, that's the reason behind why we have to complete all the antibiotics.
I see. And
Yeah, every time my wife, last time my wife, right, eat antibiotic after 3 days, right, she stopped. And after that, I scold her. I said, you have to complete it, because if you don't complete it, then the thing will come back, and then at that point of time, you will not be able to tolerate that using this antibiotic. And one more thing that is very, very crucial is that the first time when the doctor gives you antibiotics, that antibiotics is like amoxicillin, sometimes it's penicillin, right? All this is I consider as very mild antibiotics. The side effect is there. Okay, sometimes you can have diarrhea, sometimes you can have so headache, right? All these side effects, but it is not as strong as the one that will be prescribed to you if you have the resistant bacteria. Because I have a friend who took amoxicillin and all this, but it doesn't work. So the doctor need to upgrade, okay, up level the antibiotics, and then the second batch that he consumed also didn't work. So you have to go up another one level, and the third level antibiotic is really, really very, very bad to the body, right? So I think he mentioned he suffered from a lot of side effects. Yeah.
Thank you, Dr. Wenda, for the explanations. And just maybe my last questions. In terms of the carcinogenic mechanism, you mentioned that it can be a transformation from the pro-carcinogenic to become carcinogenic. So actually, how it could be transformed,
okay.
from the
So for that,
pro-carcinogenic.
maybe for that part I didn't explain it very well. Pro-carcinogenic means that the chemicals that are like by itself is not carcinogenic, but then when it is going into our body, it starts to undergo stage 1, stage 2 metabolism, which resulted in carcinogen, right? For instance, like the example that we see just now is PAH, right? PAH is I can just roughly go through. Yeah, this one. So we start out with the PAH, right? This is actually not very carcinogenic. Okay, it is considered as still okay, but when it goes into our body, when we have stage 1, stage 2 metabolism, stage 1 usually is oxidation, right? So when you have oxidation, you start to have this epoxide ring. So this epoxide ring chemical is actually a carcinogen, right? This is very toxic. So this is like activation of the initial chemical to make it into a carcinogenic chemicals. So this is what we call as a pro-carcinogenic chemical. This one. Right? Because it can be activated to produce carcinogen.
So it means it requires some activators to change from the
No,
carcinogenic.
our metabolism itself will
I
activate.
see. So through the metabolism, yeah.
Yes, through the metabolism.
All right. Thank you, Dr. Wenda, for the explanation. So to all students, since we are already over on the time, and then the student need to move for the guest lecture with problem at 9. So Dr. Wenda, I guess we can, how to say, terminate this lecture for today, and then how to say, next week we will continue the lectures. And for all students, if you still have remaining questions,
Sorry,
please, Dr. Wenda.
Yeah, for next week, I will allocate more time for our discussion. Maybe I will go through a lecture for like 1 hour, and then after that, the other half an hour, we can actually discuss through all the questions that you have from lecture 1 to lecture 7.
All
so
right.
Yeah, hopefully the students can prepare some questions, and if they have some doubts, then we can actually answer during that time.
Yeah, I guess that's a great idea, Dr. Wenda, for next week. So to all students, please kindly prepare your questions. I also still have some notes, but since we are limited by the time, so I guess we may how to say, close these lectures. And again, thank you very much, Dr. Wenda, for your great preparations for today's lectures. And I do really hope we can see you again next week. All right.
Thank you.
Wish you a great day, Dr. Wenda. And maybe we can discuss through WhatsApp, for the draft or something. Yeah,
okay.
I'm working on. Okay. Thank you very much, Dr. Wenda. Wish you a great weekend. See you. Bye-bye.
See you.
Yeah, see you then. Okay.